Hi, my name is Shamini Ayyadhury, and today I'll be talking about the work that I've been doing for the past 2 years as a postdoc in Toronto. So, biological pattern discovery in GBM using computer vision. So, GBM is an adult glioma with the IDH wild type gene, and it shows abysmal prognosis. And what I propose is that if you can understand the architecture of GBM, you can understand its function. And it's kind of influenced by this quote here, or the quote encapsulates the concept that I'm trying to drive, is that why don't we find mermaids or multi-headed dragons? And that is that structure function is limited despite heterogeneity. So in order to test this concept, of course, the first thing is we needed a large imaging data set. And I was lucky enough that I had collaborators who had done this imaging study previously in their paper, where they imaged glioma stem cells from patient-derived tumors over time. So these were imaged from 4 to 12 hourly intervals using live cell imaging. So I utilized 17,600 of these images for my analysis. And these phase contrast images were first analyzed visually by the eye before passing them through a computational pipeline. So when you look at these images, you can immediately, of course, appreciate the single cell heterogeneity in their morphometry and texture. But something that I think we kind of underutilized and underappreciate is the patterns that form as the cells start to grow and become confluent. So these are cells that are confluent and they form these directional patterning, and they are compact and they are aligned. And then some other cell types actually form these confluence or congregation points. And these structures and patterns that we find has functional significance. It's a product of their molecular property. So of course the next thing is to go through the computational work and to uncover what I call pixel associated biological profiles, and if they exist. So I used hand engineered mathematical algorithms that were already present from Cell Profiler, and then I compared them with bulk transcriptome data sets for the gene signatures, and to compare it with the pixel features that I had. And we moved on to look at the representation. So the nature of the technical and batch effects that were present in the data set, I had to split them up into 9 batches. And I performed principal component analysis across all these 9 batches, and these 9 batches are purely images that have been passed through Cell Profiler, and these are the pixel features that I've reduced by PCA. And then I compared each of these components to the 104 gene expression signatures from our bulk transcriptome dataset. And what I found is that principal component 1 and 2 highly correlated with gene signatures, with PC2 having the stronger correlation. But what we also found is that I noticed that across the confluency groups, there was an agreement in this gene expression alignment. So in summary, the high PC2 images were from samples that had higher mesenchymal microglia and injury response scores signatures, while as the images from the lower PC2N were enriched for neurodevelopmental and brain cell type gene signatures. So of course, the idea is not just to classify, like I said earlier. We wanted to understand, what kind of patterns are you observing. So in summary, what I observed is that the lower PC2 images, which were of course enriched for neurodevelopmental and brain cell type gene expression patterns, were actually more ordered they were more complex and they had smaller repeating patterns. Whereas the images that were from the mesenchymal, from the injury response arm of the samples, they were kind of more disordered, and they didn't show any unique structural patterns. The structural patterning was larger, and the entropy was larger, meaning that there was more disorder, more pattern breaking. What do you call it? Patterns were observed. Now, to complete the analysis, I did do a single cell image analysis, and in summary, what I did find was that when you just randomly choose these images and you segment single cells from them, they don't actually separate by a biological spectrum, but they do when you actually just look at the lower confluency images, the cells from the lower confluency images, and cells from the higher confluency images were surprisingly not separating by some sort of a gene expression based biology. So we can talk more about it if you guys are interested and come over to my poster. But in summary, there is a biological relevant patterning that exists despite stochastic noise. And right now, what I'm moving on is I'm moving on to understand the structure form architectural principles using spatial technologies that are available. And I'm investing in the time to develop interpretable computer vision and AI models using the concepts that I have shown you in this presentation. So that's it I have for now. I'd like to thank you for listening, and please stop by my poster to discuss, of course, to discuss science, mermaids, dragons, and more science. Thank you.